Thank you, Daniela. First of all, it's such a pleasure to stand here again. It's been a long time you haven't seen me. And it's a great pleasure to stand here after Dr. Evelyn Bishop. I'm a great admirer of her work and everything she's doing for the field of longevity medicine. So here I am again, talking about Biome, but this time I won't be rushing. Actually, I could talk about this topic for hours, because the gut-brain microbiota axis is such an important communication channel in our lives, and we are not talking enough about it. Even when we sleep, it's working. It never stops. And it's literally affecting all of the processes, all the biological processes, the psychological processes, how we are feeling, and it's a key determinant of states of health and disease. I've been asked the question, well, at Biome you're focused on IBD, and how does that actually relate to the brain? I would like to use this chance to talk a little bit more about the importance of our mental health and the status of our mental health and how it actually affects aging processes overall. Because there have been studies showing that mental health conditions actually precede the onset of biological deviations and development of certain conditions. So we really need to talk more about it. And in that sense, when we look at microbiota, gut, and the brain, it's a communication channel that is continuously interacting. It's interacting with the world around us, and it depends on all the different compounds that we are ingesting throughout the day. So in that sense, it's not just about the drugs. It's about the food. It's about the quality of the air. It's about the supplements we are taking. And at Biome, when we are talking about it, we are really talking about it at the level of information. What is the informational input that we are providing to our bodies, and how is this information further shaping who we are? And even when we look at medicine, we can see that science is recognizing the importance of gut microbiota role in this. We are only beginning to understand all of those mechanisms and how is the gut microbiota composition related to how a certain drug will be absorbed, whether a person will even respond to a drug or not. And when we are talking about the problem of responders and non-responders, we also need to talk about the numbers. 30 to 50% of patients do not respond to the drugs they are given. In oncology, those numbers go as high as 40 to 70%. 70%. When we are talking about IBD specifically, we go back to 30 to 50, but then again, we need to take into account that some people will respond to the first treatment, but then not treatments over their lifetime. And as I already mentioned, they will be sick for approximately 3/4 of their lifetime. But actually, if we succeed at what we are doing, for much longer. There are also examples of drugs that you may have heard about these days. So also metformin and GLP-1, there are studies confirming that the gut microbiota composition is a determinant whether a person will respond or not. So just to put things into context of what we have been talking about the last few days. And in that sense, at Biome, we are building an algorithm. And this algorithm, as I already mentioned, is not just about the drugs microbiota interaction. It's the drug-drug interaction, bioactive-bioactive interaction, bioactive drug, drug food. You know, how many times have you heard take a certain drug with food? But what food? Do we know what's inside that food? What time of the day? There are so many information determining how a certain drug and if it will be absorbed at all. And for that, we need this algorithm. And that's the purpose, that's the vision of Biome. And when we are talking about it, when we are talking about adjunct therapies, we are not talking enough about them, but they can completely revolutionize the field. And just imagine the vast potential of turning all of those non-responders into responders. Not even monetary, but the quality of life of people, how it would influence them. And even on the mental level, just knowing that a certain drug would work for you because we understand what is happening within your body. Our end-to-end solution integrates longitudinal diagnostics because what is lacking in the field of gut-brain axis is longitudinal monitoring. It is not enough to do a stool test every 6 months. Our stool is changing on a daily level. There are even studies showing that if you take the same stool sample and send it to different laboratories, you will get different results. So the future of understanding our gut-brain axis depends on longitudinal monitoring. And again, the intervention. So when it comes to longitudinal monitoring, I would like to now talk a little bit more about our analyzer. So it is a medical device for multispectral imaging. We basically built the technology to bring multispectral cameras anywhere in the world. It fits in my palm, and in future versions, it will be even smaller. So with Analyzer, we can already see 64 times more data points. We can analyze different wavelengths. We brought the multispectral technology into this simple device and expanding into hyperspectral as well. And we have built predictive models so that we can assess the state of one's tongue, and we can already detect gastric cancer, IBD type 2, and acute inflammatory states diabetes type 2, and acute inflammatory states in IBD. And there's numerous studies already confirming the oral manifestations of various conditions. Here are just some examples specifically for IBD, but the tongue has been a biomarker of health and disease for thousands of years through all the traditional medicines, Chinese traditional medicine, Ayurvedic medicine, Japanese medicine, and so on. And we are bringing that in a simple, scalable format that can be integrated into any healthcare system, into any clinic, so we can truly map what is going on within us, because that is the ultimate frontier, to understand these interactions. And when we are talking about gut microbiota brain, it's not just about the bacteria, it's about the human cells, the viruses, the fungi, and assessing what is happening at all times in interaction with different compounds. We have also been recognized by EIT Health as one of the top 8 companies when it comes to recognition of acute inflammatory states in IBD patients. It's a real problem, and it can be identified just based on tongue photos. We use the data that we are collecting together with dynamic biopsychological questionnaires that are accessed to our app. The app serves as an interface, and basically you answer a set of 5 questions daily. We have a pool of more than 1,400 questions, and then we are assessing the correlations. We are building, as Evelyn already mentioned, digital twin. It's a very hyped word, but we use all of that data to then understand the interactions and which compounds a person needs to activate a certain chain of reactions. Now, when we are talking about the gut, what's also really important to understand that it's not just about the colon. So we need to understand the complete system, and it starts with the mouth, the stomach, the small intestine, the large intestine. So the AI models that we are building are built around the complete ecosystem and the chain of reactions that is occurring within us. And then what is the outcome? What does the gut produce? And how does that affect us in our daily lives? Already last year, I showed these results here, so just wanted to quickly go through that as well. So we've already validated through colon on a plate study that our H-biotics, the compounds restore gut barrier integrity, and they also promote an anti-inflammatory response. But we went much further than where we were last year. So in the meantime, we have finished our first human intervention trial. So no mice, real people who are taking the product. We finished 3 preclinical trials. We have ongoing human intervention trial, 2 preclinical trials, and we are starting with 2 more clinical trials by the end of the year. So when it comes to the patient journey, we brought it in a format that is very simple and scalable. In the very beginning, we have a baseline evaluation through medical records and our proprietary digital scan. And then we start through the app with a longitudinal dynamic assessment of the gut-brain axis. And based on our AI models, we are selecting the optimal compound composition for the patient. And we are looking at the interaction between the gut microbiota and the drug. What is, again, really important is the longitudinal assessment so that we can track at which point the adjunct therapy needs to change as the person's needs are evolving. So to put this into context, we need to look at the gut microbiota axis as both a sensor and a production unit. So it's basically the most complex computer, and it's already within us. And no one is really talking about it. I think there were only a couple of talks about it here at ARDD, and it will be an enabler for the longevity industry. Because when we just think about the potential of the longevity drug, all of us here, every single one of us, is a patient. Because we are all aging. We are all getting older as I'm speaking about it right now. So the potential is massive because every single one of us is unique. Every single one of us has a different gut microbiota brain axis, and we need to understand that. And to make the longevity drug work, we need personalized adjunct therapies so we can make it more efficient in people. So our first use case is inflammatory bowel disease. As I already mentioned, what's really important when talking about IBD, people get diagnosed between 18 to 35 years of age. They go on to live for 3 to 3/4 of their lifetime with significantly diminished quality of life. And there is no cure. There are only ways to manage it. And it's really the microbiota holding the key. There are studies showing that it's the microbiota composition, specifically butyrate producers, that differentiate responders from non-responders in IBD. And in that sense, we have already validated H-biotics. So as I mentioned during the pitch, if you were here, we've seen a 3-fold increase in Faecalibacterium prausnitzii after a single dose of H-biotics. Now, what's really important is that we are not talking about probiotics. We are not planting the praus within a person's system, because that's kind of like, oh, I'm going to throw a seed here. Will it grow? Nobody knows, right? So what we are doing, we are using this channel, and we are modulating it, bringing it the compounds it needs so that it produces what it can already produce. So we believe that the system is already intelligent enough, and that actually when we have states of various diseases, we can impact it so that we alter the composition so that drugs can be more efficiently absorbed or absorbed at all. What's really significant are the results of our most recent gut-on-a-chip trial in IBD donors. What we've seen is an increase in butyrate production up to 400%. So 400% butyrate production increase in IBD donors. And we are now looking for a pharma partner to take this to the next level and to conduct a pilot study and to see the interactions in real people with real drugs. Now, when it comes to short-chain fatty acids, again, they're a very important topic. They regulate many mechanisms in our body, from the way we feel, from the way we behave, even our ability to make executive decisions. They control our appetite, energy intake, so such a variety of functions that are defined by the information that is produced within the gut. And they're also connected to muscle mass. So another important topic, I think we heard quite a bit about it these days. So short-chain fatty acids are also an important marker when we are looking at muscle mass. And there are studies showing that there is reduced fecal butyrate in elders that have lower levels of muscle mass. So again, something to consider specifically when it comes to inducing short-chain fatty acid production. At Biome, we work with global academic and industry leaders in the field. As I already mentioned, we finished our first human intervention trial. We have multiple clinical trials going on. What's really important is the translation of all of these findings. And I would like to emphasize that we are using the data from the studies to further build our algorithm, to further build the understanding of the gut microbiota-brain axis. And everything I just mentioned is in that corner up there. So basically, there's still a lot more upcoming, including understanding how do abiotics influence postprandial glucose response, and further validation of the multispectral imaging in clinical settings. We are building a robust and diverse IP portfolio. Besides the data from the studies, we also have exclusive longitudinal data from people who are already utilizing our products. So we have a community of members that are using Biome, that are taking photos of their tongues daily. It's actually really fun. So if you become an Biome member, you will get the opportunity to take your photo of your tongue daily. We built the largest tongue data set in the world. We are continuing now, as I said, with clinical validation of the multispectral imaging. We have proprietary trade secrets and patent-pending medical device, digital diagnostics, personalization engine, as well as the molecular compounding method. You may have also heard yesterday on the panel my founder Bruno, who is here. So if you'd like to discuss Biome and how it could potentially integrate, or how do you perhaps see some potential business opportunities, we are here. Do approach us afterwards. And we truly believe that the longevity drug will be an algorithm, not just a molecule, because we need to understand all of these different interactions. And we see the role of Biome becoming the NVIDIA for longevity, enabling the understanding of the gut microbiota-brain axis so we can personalize adjunct therapies, because, as I said, when it comes to aging, we are all patients, all of us here. So the potential is massive. And as a very last thought, I would like to use the opportunity to give special thanks to Longevity, who they have backed us, but not just that, but they have shown immense support in helping us to get there, to get where we are now, and especially in development in Medical and building our scientific advisory board. So guys, I'm really grateful for all the support you've given us. That would be all. Thanks a lot.
Thank you so much. We have time for a very quick question.
You very nicely showed that the microbiome is a very complex multivariate problem. So I was wondering then, for why do you go for your proof of principle for a univariate biomarker? So it's very cool that you can change the microbiome, but you change one biomarker in the IBD. Why not do 10?
Well, first of all, this is just a beginning when it comes to our studies, when it comes to IBD specifically. So we were not only looking at butyrate, we were looking at other short-chain fatty acids as well. So we've increased overall short-chain fatty acid production up to 300%. And we are continuing with metagenomics and metabolomics to understand host-immune interactions. We will be also looking at tight junction, what was the impact, and so on. So it's still the first results, and we will continue further in that sense.
Okay, thank you very much. Thanks for joining.